Hello to everyone. A very warm welcome from me to this webinar on NRF2 and system medicine. I'm Luciano Saso. I'm a professor of pharmacology at Sapienza, University of Rome, and it is a great pleasure to see you all today. This meeting is part of a Redox webinar series organized in cooperation with Mauro Maccarone from the University of L'Aquila, Alessandro Giuffre from the National Research Council in Italy, Marzia Arese and Elena Forte also from Sapienza University of Rome, and Alessandro Leuti from the Campus Biomedico University of Rome. In particular, this webinar is organized in cooperation with the Coast Action CEA 20-121, entitled Bench to Bedside Transition for Pharmacological Regulation of NRF2 in Non-Communicable Diseases, which, as most of you know, is coordinated by Professor Antonio Quadrado from Autónoma University in Madrid. So I would like to thank very much Professor Quadrado for the promotion of this webinar and for the very nice cooperation that we have. I also want to thank Brigitta Buttari from the Istituto Superiore di Sanità in Italy for the organization of this webinar, and the distinguished speakers that we have today, and all of you, dear participants, for joining us today. So without further ado, I leave the floor to Professor Quadrado, who will chair this meeting today. Thank you and have a good webinar.
Thank you very much, Professor Luciano Saso. I think the reason why we are organizing this specific meeting on systems medicine is because I think it is important that we change the scope we have about disease, changing from symptoms to pathomechanisms. I learned this several years ago thanks to the collaboration with Professor Harold Smith, and I am sure that he will go deeply into this aspect of systems medicine and also how we can use our knowledge in order to repurpose new drugs. Then going more into the subject of the cost action in which we are involved, Professor Tamas Kostinaros will present how NRF2 is connected in this network of chronic diseases, probably the layout in which this transcription factor is participating. And then we have George Spirou, who will give an example, specific example on the network in which NRF2 participates related to Alzheimer's disease and drug repurposing. So without any additional explanations, we will go now to Professor Harold Smith. Harold, I guess, is known by many of us. He is a professor at Maastricht University. I don't want to take a long introduction because I think most people know him. So Harold, please, now the floor is yours. Thank you.
Yeah, thank you very much for the invitation. A lot of what I'm showing is also part the results of another cost action, which I led together with Antonio, just that was focused on ROS in general. So let me start first why we and many others think we need a new approach to disease, but also a new approach to ROS. The approach that I'm going to talk about is about this new type of medicine systems or network medicine, where we are identifying disease mechanisms by a completely new way, no longer by canonical pathways, no longer by single targets. And of course, all of this is only relevant if we can validate it in patients. So I will also talk a little bit about that. So these are my collaborators. Down here is this cost action Euros, where I worked with Antonio. I then led another one on systems medicine, currently leading a REPO trial, which is systems medicine-based drug repurposing. And I've now the honor to lead a 25 million euro project to build a platform for network medicine and drug repurposing. So what you very often read about oxidative stress causing disease. Actually, if you Google, I would think there is almost no disease that has never been associated with oxidative stress. So something is funny about this disease mechanism. Also, there has been for many years this quite simple scheme. A lot of ROS is bad or too little antioxidants are bad. If you have a lot of antioxidants and very little of ROS is good. That concept, has its shortcomings because there are clearly a lot of essential functions that require a lot of ROS. So a lot of ROS cannot always be good. Then this concept was slightly changed to causing eustress or distress. So I think the problem this simplification has is that ROS are not only there to cause damage. So that's why the simple approach of antioxidants never really worked. The thing is, one has to identify which are the sources of ROS, like classical pharmacological targets. What are the enzymatic sources of ROS? And then one has to acknowledge that there are also several processes that generate ROS that have signaling function or synthesis function, for instance, thyroid hormone synthesis or immune defense. So ROS are not only something that needs to be eliminated from the body. And what determines disease sometimes can be the quantity, but sometimes it can also be just the location where ROS is formed, that the location, either cellular or subcellular, is an unphysiological one, or that the type of ROS is changing, let's say from hydrogen peroxide to superoxide. So the alternative approaches are also to take degradation into account, endogenous degradation. So with this, one can think of inhibitors that inhibit those sources of ROS that drive a disease, or that are able to repair a damage of ROS. I will also show you one example of that. Or to activate an endogenous sort of ROS degrading mechanism that is beneficial. However, this has the risk that the same mechanism that you are activating may also in other cells that are not involved in the disease in the moment induce an unfavorable degradation process of ROS. The problem is when you take all the sources and targets of ROS together, they are usually summarized in Wiki Pathways or KEC pathways. And basically the assumption is that everything that generates ROS, metabolizes ROS, or is a target of S, that you can always simplify it by ROS. So no matter where the reactive oxygen comes from, as long as you deal with it, you basically achieve a positive effect. So that is quite unlikely. The other problem that Antonio already mentioned, when we think about where to which disease apply ROS-based therapy, is that we currently hardly understand any disease, if we're honest. Most diseases are diagnosed when a patient, out of a state of well-being, comes to a doctor with a symptom, or the doctor measures an abnormal parameter like elevated blood pressure, elevated cholesterol levels, and so forth. And then the disease is typically named after that symptom in an organ. So asthma, hypertension, psoriasis, inflammatory bowel disease, these are all descriptive terms, but they are not a molecular definition of a disease. So, and with such imprecise descriptive disease terms, the only thing you can do is you can treat symptoms because you do not know the cause of the disease. And if you can't repair the cause, you end up automatically with a chronic disease. If you can't repair something, it will be chronically defect. That's why we have so many chronic diseases. There are several reasons for that go wrong in the moment in research. But one of the key conceptual error is that we have split up the human body organ by organ. And so, for instance, there's the neurologist, Department of Neurology and Neuroscience. There is Department of Cardiology, the cardiologist, and cardiovascular research, and so forth. So we really believe we can look at one organ. Whatever we see there is the disease. Whatever the patient has in any other organ cannot have anything to do with my disease. This must be another disease. I defer it to another specialist. Let him deal with it. We are working in the clinic and in research in absolute silos. And in a moment, the big thing is, to digitalize things. But if we keep everything the same and just do it digital, we won't change anything. So we really need a paradigm shift. And for me, the eureka moment was when I saw this paper from Laszlo Barabashi. That's a network scientist. He looked at all diseases, all human diseases. And the color coding here of the diseases is this organ-based nomenclature of diseases or taxonomy of diseases. And in this network, he linked diseases through shared risk genes. So, for instance, here in the middle, there's a gene which is a risk gene in 3 diseases. So this gene links then somehow 3 diseases. And if you take all diseases and all risk genes, you end up with this network. And you can see that this network is not evenly distributed, but some diseases cluster together. So they form little groups and they cluster together because they have a lot of common risk genes. So the risk genes are basically the glue between them. And you will also see that many of these clusters are multicolored, which means diseases which have symptoms in different organs are dealt with different specialists, are researched in different research disciplines. They actually belong together. Actually, this concept is not such a paradigm shift. If you think of rare diseases, which are very often caused by a single gene, but in this case, we know the cause of the disease, a single gene. Even in rare diseases, the patients very often have 2, 3, 4, 5, have symptoms in 2, 3, 4, 5 organs. So if we would not know that this is a rare disease and caused by this single gene, our current medicine could easily make out of this 3, 4, 5 entirely different diseases. So actually, from such a network to the paradigm shift, it's not a gigantic exercise. Basically, the layer that we look at it now of the current disease terms, those we just declare the symptoms, and the genes below those diseases or symptoms that actually bring those clusters together, they will become the molecular causal disease definition. So we will do the same thing we do with rare diseases, but the new disease definition will no longer be organ or symptom based, but it will be based on molecular mechanism. That's the approach. So of course, these networks are not 2D, but actually 3D best described. You can put on virtual reality glasses and then dive in, look into your favorite disease, look which genes are around, or you look at your favorite gene and look which diseases are around. We work on this cluster here, which when you look at it, you think, well, that's quite a mixture of diseases, you know, diabetes, stroke, heart failure, asthma, Alzheimer, hypertension. What have they got together? Well, they share a lot of risk genes, and a lot of drugs are actually active in all those diseases, in several of those. And of course, for not all diseases, we don't know all genes in the moment because of the lack of genetic studies, but you can also form these disease networks by other manners. For instance, comorbidity. If two diseases very often occur together in the same patient, they may actually be the same disease causing symptoms in two organs. Or if two diseases have this almost the same symptoms, maybe there are not two diseases, maybe there are the same disease causing symptoms in several organs, and so forth. So we're not ghost drivers. There is Christina Kiel in Padua. She is working on retinopathies, and she has split them up in 3 distinct disease types. There's a group in Kiel in Germany that works on autoimmune diseases like rheumatoid arthritis, colitis ulcerosa, Crohn's disease, and so forth. And they say we should actually no longer use disease terms like rheumatoid arthritis, colitis ulcerosa, psoriasis. They are completely misleading. They suggest they define a disease, but the only thing they do is they describe a symptom. And there's also a very nice paper from Sanjay Swiga and Cell, where they delineated all human cancers down to 10 cancer pathways. The only shortcoming is we are not convinced of the use of CET pathways. I showed you that before. So probably we have to further subdivide them in several more modules. But the principle to define cancers by independent pathways into lower and higher complexity cancers is a very nice paper to look at. By the way, I've sent around a handout, so you can look at some of the slides afterwards, if you like. So in this cluster of diseases, we found the following genes. Some were related to cyclic GMP signaling, some were related to reactive oxygen signaling. And if we would now use CET pathways, that's what geneticists usually do. They annotate genes that they find to CET pathways. We would end up with this CET pathway in green and the RS pathway in red. Altogether, close to 400 proteins. What a daunting result that you have a cluster of diseases and now you're supposed to potentially deal with 400 proteins. So this could not be the case. So we had a different approach. We wanted to define this disease pathway differently. And we started with the human interactome. These are all known protein-protein interactions that we know in the cell. And we took those cyclic GMP genes or their proteins as seeds. And then we looked, what are their first neighbors, their direct first neighbors in the interactome. And from all these ROS potential proteins, the only one we found was NADPH oxidase 5. There's also NOX1 and NOX4 here, but if you look closely, they're not directly connected. They are second neighbors. That's one method. There is another method where you first form the modules and then look where are your proteins. That's called global modularity optimization. Again, NOX5, the only other neighbor. And then there is yet a third method, Steiner trees, and again, NOX5. So it looked like out of all these hundreds potential proteins, we could focus primarily on one. So basically, these CET pathways is one key message. I think to identify disease mechanisms, they are completely useless. They are a nice mind map if you want to learn by heart everything that was ever found about RS or cyclic GMP, but they have nothing to do with disease-relevant pathways. So only those elements were actually sufficient. We then also looked at all cyclic GMP, sort of basically to split up the cyclic GMP pathway, but I think for you it's more interesting to split up this RS cake pathway into RS modules. So we found separate NOX1, NOX2, NOX3, NOX4 modules. This is the module where we found NOX5. This is not a pure RS model. It contains cyclic GMP genes as part of that. So it's actually a heterogeneous module. That's another message. There are no clean pathways. You know, you always find other proteins in there. Then the enosyntase model, NOS1 and NOS2. NOS3 actually is in here in the ROC module. Monoamine oxidase, then two separate modules for MPO and xanthine oxidase, and your main interest, the NRF2 module, which we later together with Amber Gune and Antonio worked out in a little bit more detail. So, which means these are independent signaling modules. You cannot easily just merge them together, so construct connections between those. A priori, they are separate modules involved in probably separate diseases or maybe no disease. If they are never associated as a causal gene, then in no disease. Yeah. So and that fits very well with the Nature paper that came out this year, which focused on, I think they looked at thousands of images of cells, and which highlighted cellular organelles. And what they found actually, that organelles are not randomly distributed within the cell, that they actually form kind of like organs in a cell. And these sections are comprised of little modules. So that's exactly what we find. And you can't have these gigantic pathways in a cell. There just is not the space. The cell is compartmentalized and interconnected small signaling modules, which you have to identify, and they are then the disease mechanism. With respect to this NOX5 story, just a quick validation. NOX5 is a gene that's not present in humans, so we did a NOX5 knock-in mouse, we let it age, and so we were pleasantly surprised that these mice became hypertensive, because hypertension was one of the phenotypes in this cluster. And then when we looked why are they hypertensive, we did acetylcholine-dependent vasorelaxation, which is a cyclic GMP-dependent mechanism, and that was impaired. That's the red curve in these NOX5 knock-in mice. So not only did the phenotype match, but also the mechanism by which the mice became hypertensive, namely impaired cyclic GMP signaling, also matched this disease module. So, of course, we are not naive, and we think we can explain all forms of hypertension now, but where this one mechanism, but only a pizza slice. And the other forms of hypertension, for those, we need to find other mechanisms. So what we're now doing, we take out the patients with this symptom, hypertension. So these are all those patients. Then with the diagnostic, we pick out those patients where we can really identify, let's say, upregulated NOX5 or impaired cyclic GMP signaling, and only those patients are then included in a mechanism-based clinical trial with not just antihypertensive drugs, but drugs that specifically target this disease mechanism. So different drugs, not drugs that are typically used in hypertension. And this shows the stratification. This is the distribution of NOX5 levels in green in normal patients. and the red in hypertensive patients. And you can see that most hypertensive patients have fairly normal NOx5 levels, but there is a subgroup of hypertensive patients which have clearly elevated NOx5 levels, and only those patients are treated. Those patients with hypertension, for those we need to identify another mechanism. And heart failure with preserved ejection fraction is another tough nut in the moment in the cardiovascular field. And here it was also part of our disease cluster. And here we found again a subgroup of patients that is above this pathological threshold. It also forms a subgroup, and that's the trial that's now starting in Valencia this month. First mechanism-based curative treatment of heart failure with preserved ejection fraction. And the third trial that we're doing, that's running already in the moment, is in stroke. In stroke, the situation is slightly different. There we don't have a deficiency in NO, but because of the opening of the blood-brain barrier, we have excitotoxicity. It's because of the hypoxia. Another enzyme gets induced, NOx5. So we have overproduction of NO and overproduction of ROS. And then some unpleasant chemistry occurs in between. This doesn't really matter exactly to explain what all happens, but then all of a sudden we have unphysiological interactions. And therapeutically, there are SGC activators which sort of repair this damaged ApoSGC by ROS. So that's an example where you can actually repair damage caused by ROS. But what about those here? There is no NOS inhibitor on the market, and there is no NOx inhibitor on the market, and we want to do drug repurposing. We don't want to develop any new drugs. So there's huge potential for drug repurposing because most small molecules bind to 30 other proteins. And so we found that a thyrostatic is a fantastic neuronal NOS inhibitor, and an antipsychotic, perfenazine, is a fantastic NOx inhibitor. They are all approved ethically and by regulators to be used in this trial as NOx and NOS inhibitors. And then that's what we call network pharmacology. By targeting this network at 3 sites, we can kind of normalize it. Sometimes I hear that network pharmacology is not much different to combination therapy that we're currently doing, but it's completely different, because what we currently do, we do symptomatic therapy with several drugs which we combine, but they are mechanistically totally unrelated. And such therapies are maximally additive. Usually they aren't even additive, and they're definitely not curative. What we are doing here is we treat the causal disease network. And those targets are all mechanistically interconnected, so they are highly synergistic. And this has been shown by Anna Casas in the stroke model. So she used those NOx inhibitor, NOS inhibitor, and the SGC activator. She lowered the dose. All compounds have an effect. They all protect in stroke. But she lowered the dose to a level that they all became ineffective. And when she combined these 3 ineffective doses, she got the full protection again. So that's a perfect proof for extreme synergy. So to conclude, a lot of things also need to change in research, because this concept from basic to animal to clinic, quite frankly, we know it doesn't work. Hardly anything translates. And we want to cure patients. So we need to use the vast amount of big data about humans, patients that already exist, work together with bioinformaticians, use other multi-omics approaches, and then establish step by step a completely new disease definition, or a completely new pathology, a molecular pathology that defines all diseases by molecular mechanisms. And this may already be the lead towards a clinical intervention through the high potential of drug repurposing. the real research need we see sometimes is to develop the diagnostics that are able to pick out the patients, that have not only the symptom but the mechanism. And I think the need for animal models, there was this NOX5 knock-in model, this humanized mouse, maybe in stroke, these acute scenarios. But I think the role for animal models, until other than they perfectly mimic the human disease, is highly questionable. And with that approach, we will be able actually to cure diseases. And if we take prevention more serious and early disease detection, we may very soon be able, with biomarkers, to first detect the genetic risk in the patient, monitor whether the risk evolves, and then intervene even before the patient has ever experienced symptoms to actually reverse the disease, so that in the ideal scenario, the patient will never experience his or her disease symptoms. And if you're interested in that sort of this network medicine and this drug repurposing, the next conference on that will be in October in Stockholm, the year later in Washington, and then 25 in Madrid. Thanks very much.
Wow. Amazing. So I guess it was, thank you for this very provocative lecture. I hope you will have some interesting questions at the end.
Yeah.
If not, for sure, we will think a lot about all these interesting ideas that you provoked here. Okay, now we are going to move to the next speaker. It is Dr. Tamas Korksmaros, and he's going to talk about the role of about this specific network that is related to our PET transcription factor called NRF2. Please, Tamas, whenever you want.
Thank you very much. If everything goes well, you can hear me and see my screen.
Yes.
So thank you very much for the great opportunity, and especially it's a privilege to talk after Harald and after all the lots of really important points that he already mentioned. So I will try to continue from where he left, but with some specific examples. So just a brief introduction regarding my group. Which is basically currently based in Imperial College London. In the last 8 years, I've been working in East Anglia, in Norwich, and my group is basically working with omics data analysis with different computational approaches, as I'm going to show, and we also do some validation that I'm not going to show today. And in principle, we are interested about cell-cell and cell microbe interactions in the gut, and its effect on precision medicine that I'm going to mention. And as you will see, my main interest is in signaling networks, and then we did multiple projects around NRF2 and the antioxidative responses I'm going to show. This is my group, which is a really nicely multidisciplinary team, from computational people to real hardcore experimentalists and a couple of clinical researchers as well. So as it was mentioned, network medicine was, or network pharmacology was mentioned before. I just wanted to show this slide about network medicine, which was coined also by Albert László Barabási 15 years ago. And there were a couple of other points made about network medicine, how it could work as a think globally, act locally type of concept. And as Harald mentioned, that basically there are now multiple ways how we can use networks as a tool to develop better therapies, to repurpose drugs, and to stratify patients, as I'm going to mention in the last bit of the talk. But before we jump into that, why do we need networks, and what we can do with that? And in a way that can be considered as a kind of an address to the very, very correct issue that Harald mentioned about KEGG, for example, which is a very popular pathway resource, but it's very limited, as he pointed out. So when we are just considering pathways, and this is obviously just a model of that, then we have different ligands and receptors, and different mediators take the information to transcription factors. And these pathways indeed can be very complex and they intertwined. There are crosstalks between pathways. And it's very relevant that this is how they work, even if they haven't evolved like that. These crosstalks make our system very complex, and this is how a very low number of proteins can be responsible for such a diversity and stress response that our cells are capable of. But network of signaling pathways is not enough only for that. There are also additional connections and networks between transcription factors, so interactions between transcription factors, which are often not actually directed, they are protein complexes. And then these transcription factors have their target genes, and those target genes can actually encode specific genes or microRNAs, for example, that can downregulate the processes. And then this is also a very important feedback loop. And establishing a more comprehensive view of how signaling is working is quite important, because, for example, just imagine that there's an incoming signal from where I'm showing it now, and you see that there is a change on this protein, while these two are not connected. But properly mapping the signaling processes, we can actually see that there is an incoming signal that goes to the transcription factor that is actually responsible for the expression of this other protein. So these feedback loops are very important to understand the dynamics of our signaling processes. And we would like to build these networks. We would like to understand how they work. And with the advance of multiple omics approaches, actually we have lots of data sources and experimental methods to collect information on that for a specific pathway. A good thing is that there are also existing resources, quite a lot of them, that contains very specific information on parts of these networks or the components of these networks. The bad thing is, however, that there are too many of them. And in order to get a more comprehensive global picture, probably for a single project, you would need to use at least 10, 15 resources sometimes, depending on how many different interactions you would like to use, which is often time-consuming, limiting, and often can lead to lots of troubles, because these resources, unfortunately, not always meant to be integrated with the others. So realizing these issues, my group has been working in the last 10, 12 years on developing tools to integrate these data sets properly. And one of the flagship projects for that was the signaling resource, where we basically demonstrated the concept that we can have a manual curation from the literature, which can be multiple different signaling pathways or other processes that I'm going to show. And then from many other resources, we can connect relevant protein regulators, transcription factors, and microRNAs to this network to again, in a single place, like the signaling.org website, to access these resources. And you can look at it by proteins, so you can easily access a single proteins interaction. But here, this is the screenshot of a download page where you can select the species of your interest. Actually, now there's a zebrafish as well, the pathways of interest, and the different regulators regulating the inner layers. And you can download it in different file formats to open it in Excel, CSV, or Cytoscape, where you can directly visualize it. And lots of people, luckily, are using this resource, around 1,000 people per month. And just last year, we published a new version for that, where you can filter these interactions based on tissue and cellular compartment, which is, I think, a very important point in systems and network analysis. Otherwise, we are aggregating lots of information. So that's, I think, a very, very good thing there. And also, we doubled the number of pathways with lots of immune-related pathways there. On the right side, you can see this protein page where you have the information on the protein, a single protein, and then you can look at different types of interactions. And a novelty in this new version is that you can just download that for a single protein. You don't need to download the full database if you're interested about the specific signaling protein. So we added lots of other information to this resource that I'm not going to the details now, but basically for model organism, this is one of the biggest signaling network resource, and for human, it's also quite an important contributor to what we know about signaling pathways. So I already mentioned that with these integrated resources, we can integrate different types of interactions. Now I just would like to term these multi-layered networks, define them. So the good thing with that is that you can have different types of networks. And as I mentioned, different omics approaches can provide information to that. And there is this very interesting one, which I will talk about in the final part of my talk, which is the genomics part, to make patient-specific networks. The point is that with different modeling approaches or just understanding how these networks work, we can understand the effect of a compound, for example, on one layer, then its effect on the signaling part, if it activates different signaling pathways. And then we can predict which transcription factors are getting activated, and by them, how the regulatory network is affected. And basically, with the transcriptomics data we detect at the end, we can validate the upstream model. So basically, multi-layer networks are a really good scaffold to integrate and interpret multi-omics data, because you can get the mechanism, a predicted mechanism for certain outputs. After this introduction, I just would like to briefly introduce a resource that we developed quite a long time ago, but both we are using it, and luckily the community is very heavily still using it. So we were encouraged, and in the near future, we are going to update this. But I just want to show how it works. So the main motivation, I'm not going to introduce obviously NRF2 to this audience, but I just wanted to mention the main motivation for a systems biologist like me, why I wanted to work with NRF2. And that was basically this fantastic functional set NRF2 can actually do as a single master transcription factor. And I was interested how it is regulated, how a single protein can actually be important in cell survival, cell death, and different homeostatic processes, and how it can be turned off, and things like that. So first, I was interested at what are the protein interactors of NRF2. And then we did a study which looked like as the following, as you can see. So we did a manual creation with the published protocol we use for the big signaling network resource I mentioned earlier, signaling. Then we looked at existing resources, and we used computational prediction of protein-protein interactions using domain-domain and domain-motif interactions. And we ended up with this distribution, which was kind of pointing out the importance of the three different types of approaches when you would like to understand the process. Because these existing resources are very good, especially Intact and BioGrid are very good big network resources containing relevant human protein-protein interactions. But often their curation is lag behind because of often just resources, manpower. So actually doing a proper manual curation is often helpful. And predictions can contain false positives. However, they can also point out interactions that can happen. It's just there were no experimental tests before to look at them, so they can guide experiments. After, by the way, publishing most of the manually curated dataset have been moved to Intact. So if you would repeat this study, probably you would get some manual curated only interactions because of the time changes. But basically, we are communicating a lot with these different resource developers, and our manual curations are feeding into their work. So at the end, we ended up with this protein-protein interaction network module, so to use this term, around NRF2 and KEAP1. So this is not a snapshot from Star Wars, obviously, but this is a nice interaction network, just pointing out the different positive and negative regulators with the green and red respectively interactions affecting the NRF2 protein by multiple signaling proteins. So basically, as a summary of how the interactome looks like, we wanted to also understand what's the output of NRF2. So we were using known binding site information on NRF2, and basically we identified more than 7,000 target genes for NRF2. And looking at the regulatory network, we also identified 34 transcription factors capable to regulate the expression of the NRF2 gene. And then the interesting thing was to look at the overlap. So we found that 3 transcription factors out of the 34 are also regulated by NRF2, which we basically call the regulatory loop. This is an example for that. This is PPAR gamma, which is regulated by NRF2, and NRF2 regulates PPAR gamma. And actually, there is a protein-protein interaction between the two as well. So the interesting thing with this figure was that at the time when we published this, all the interactions, all the 3 interactions were separately published before. However, there was no, or at least we were not able to find any evidence that someone connected it properly, that actually they are co-regulating each other. And it's not a unidirectional NRF2 PPAR gamma or PPAR gamma NRF2 regulatory interaction. So it's quite important when we are looking at these stress response processes, and we did a couple of further studies on this co-regulation, how important it is not only for stress-related activation, but also later on the inactivation of stress response. Another type of regulators of NRF2 are microRNAs that can downregulate the mRNA or the translation of NRF2. And we found 85 microRNAs that are potentially capable to do that. And thanks to putmir and transmir resources, we were able to find 164 transcription factors that could regulate these microRNAs. And again, because we had transcription factors there, we looked at the overlap between the target genes and these transcription factors, and there are different combinations. So we found more than 300 different regulatory loops. So when we did this study, just as a disclaimer, 10 years ago, and some of these resources have been updated nicely, some of them are the same. What I wanted to point out is that at the time of the publication, we checked each of these regulatory loops, each of these 300 regulatory loops, and around, if I remember well, 16 of them were known. And we published this resource, and I was very happy to see that in the following 5 years, it guided experimental studies, and we got lots of citations from colleagues who were looking at specific context and cell type specific downregulation of NRF2 by microRNAs, and kind of validated dozens of our predictions. So basically, we wanted to put together the regulatory and the interaction parts, so the regulome and the interactome of NRF2. That's what we call kind of NRF2-ome. This is basically that what you have seen before, this more than 200 proteins that can modify or bind to NRF2. And we also found around 39 proteins interacting with NRF2 as a target gene. One of them was JNK1, which can phosphorylate NRF2, as many of you know, and actually NRF2 can affect the expression of JNK1. So again, that is a very good example for feedback mechanisms that can help to turn on or off the stress response. So after we created this resource, and basically created these networks and did a network analysis on them, we wanted to go further, and we provided this for the community in a resource called NRF2-ome. And basically, the core of this resource is this manual curation of the interactions and target genes of NRF2, looking at the protein interactors and target genes of NRF2, using experimentally verified interactions and predictions, and then basically connecting those layers that I mentioned about regulations. We also added in this resource a final layer, which was basically connecting all these transcription factors we identified around NRF2 to the signaling pathway database that I mentioned. And it allowed us to connect signaling pathways and signaling processes to the regulation of NRF2. So this is just a screenshot from the website where you can look at the protein involved in a NRF2 regulation. Obviously, this is a key one, KEEP1, where you can look at individual interactions. You can look at the actual evidence for these interactions. You can visualize the network for that. In case of NRF2, there are even more interactions on the website. You can look at specifically these regulatory loops I mentioned earlier, which is, I think, a very good way to look at NRF2 regulation. You can filter with what kind of feedback loops you would like to look at. And actually, if you go to the download, currently the only option is that you can download the whole database in an Excel format. So you can get everything that we collected. And I hope that we will have some funding in the next 1 year or so to update the resource, and then with that some functions of the website as well. And yeah, these are the microRNA-based feedbacks that you can also look at. So at the final part of my talk, I just wanted to present a story, a project, which is not yet directly connected to oxidative stress response or NRF2, but it can very quickly and easily be connected, and that's our plan. So that's why I called it the next steps. And this is related to precision medicine, which I'm kind of more and more interested about. For this project, and mostly on many projects in our group, we are looking at a disease called inflammatory bowel disease, which is an intestinal chronic disease with, unfortunately, quite a lot of patients experiencing it, and then it's a chronic lifelong disease. The interesting thing, from a systems biology point of view, is that we only know that there is a genetic background for this disease, often related to autophagy in Crohn's disease, which is a type of IBD. So the genetic susceptibility connected with some kind of immune dysregulation and changes in the microbiome. But this general definition exists for decades, and we don't really know more how and why a healthy gut that you see on the left is turning to a very, very inflamed one you see on the right. And patient stratification, which would be key because there are many existing drugs available for these patients, would be very important, because in many cases patients are not getting the drugs that are working for them. So our question was that in order to understand the pathogenesis or even the treatment options for IBD, can network approaches help? And we started looking at genome-wide association studies data that has been done in the last 2 decades, identified more than 500 SNPs, single nucleotide polymorphisms, and many of them were non-coding. And this is quite important because non-coding regions are more difficult to annotate and understand what they are doing, how they have a phenotypic effect, and which cell types they have effect. So last year in Nature Comms, we published this pilot study, basically, where we looked at around 340 patients who had ulcerative colitis, a type of IBD. And we individually looked at the patient's SNP profiles. And we actually identified that most of the SNPs, well, many of the SNPs, can be found in transcription factor binding sites or microRNA target sites, meaning that these SNPs can have an effect on how these transcription factors can regulate a target gene. So in the bottom right, you can see that there's a transcription factor and there's a change in the target gene or a microRNA, and there's a change in the mRNA that it can target. And we didn't stop here, and we did a very similar study like what Harald mentioned, and I think that's a very key thing nowadays in systems biology, that we looked at the first neighbors of those proteins that are interacting with these genes encoded by the SNP affected genes. So first neighbors is a very, very good and context-specific way to expand your data set and understand better the downstream effect of any kind of genetic or other change. So with the same color coding, I just wanted to show this, how it looks like. So with different transcription factors, microRNAs and their SNP-affected proteins and their first neighbor interactors. This is how this network looks like. And then we did a functional analysis on them. And we identified very relevant functions relevant for this particular disease, which was quite reassuring. However, the question was that how these SNPs are actually, whether we can cluster the patients based on these SNPs. And before I could jump onto this, I just wanted to show you this hairball. I hate this. So this is an integrated network, a big signaling network, which where with yellow we pointed out all those genes and proteins that are affected in any IBD patients we looked at, so in the whole cohort. And this is not really useful. So how can we make with network approach a useful figure? And what we can do with such data set is that we can do patient-specific analysis. So here you have a network of a single patient, where with yellow we are pointing out those genes or proteins that are affected in this particular patient by the SNP, and with blue we are showing those proteins that are not affected in this particular patient, but in our cohort there is at least one patient where it is affected. And if we do this visualization, and we did it for all the 300 some patients, we started seeing a really nice patterns, which in parallel we confirmed with some machine learning clustering algorithms as well, that actually patients were nicely grouped to different based on a few driving proteins or SNPs affecting PKCB or SABDP regulators. There were within that main cluster, there were two subsections who were HLA related, HLA gene related, and in the other ones, it was the VEGF pathway that basically split the clusters to two. The interesting aspect with all this approach was that we identified patients which had a really big effect of their SNPs, and also some patients which had only a few SNPs, but they still have very similar clinical symptoms. But their clinical symptoms were similar. Their pathogenesis is probably different. So with this network mapping, we can understand better why patient can be different. And this could be quite important when we would like to treat them, especially when these patients are getting immunosuppressant drugs, which is really good in many cases, not for everybody, but in many cases to reach remission. But unfortunately, most of the cases, they relapse. So understanding the difference between patients could actually hopefully help to keep them in remission. And we are doing multiple experimental studies on comparing the characteristics of these patients. What I can briefly now mention is that these network footprints, as we call them, these yellow parts, can actually guide us to specific cell types. So we can identify specific pathways behind these or specific processes that are cell type specific, pointing out a cell type specific malfunction in these patient groups. So just to summarize, with this project, we are looking at these regulatory effects, which is, I think, quite important. And with NRF2, I already mentioned that these regulatory interactions combined with the interactome are quite important. We also pointed out that just not looking a single SNP, but a set of them, so the co-occurrence of SNPs is also very important. And looking individual patient data can often lead to better success than just aggregating and looking at everything. And currently we are working with the biobanks to correlate our network clusters with clinical metadata. And basically the project that is quite interesting and couple of recent reviews very nicely pointed out is the role of NRF2 in IBD, and how basically we can combine these two resources, this NRF2 resource I presented and this precision medicine patient-specific network tool that I showed. So this is what now we would like to do. And before I finish, I just wanted to summarize these computational resources we have developed, as some of them could be useful for you. So I mentioned signaling and obviously NRF2. There's also an autophagy regulatory network resource we created, and cytokining, which is a cytokine-based interaction network. All of them are similar in a way that they have this multi-layered structure. And as Harald mentioned, KEGG is not the best way to look at signaling pathways. And I'm not going to say that signaling that we developed is, however, the best to do that. But of course it is. But what we did is that together with the group of Julio Saez Rodriguez in Heidelberg, we created an integrative resource called Omnipass, which contains more than 50 different signaling pathway resources that you can access and look at. It also contains lots of other information other than the signaling processes about localization, expression, protein structures, homology translation to mouse, if you're working with mouse and not human data. And with Omnipass, you can access just the database, or there is an R, Python, and Cytoscape modules to access it from different computational ways. So I think it's a really good one-stop shop for looking at any kind of signaling related information. And in our group, basically, we are using it with different pipelines. I presented ICE, the precision medicine tool. We have 2 other pipelines that I did not have time and focus today for presenting about single cell analysis and host microbe interactions, which is a very interesting topic, actually, how these host processes are regulated by microbes. And with this, I would like to finish and just thank my group, lots of people who were and still working in my group for these projects, lots of collaborators that are supporting our work, especially Diana Pup, who was a key person in developing the NRF2 own resource. And I just wanted to mention that I'm organizing another workshop in the summer, the 3rd one of this type, where it's really a multidisciplinary workshop. We are connecting different fields with really high level, really good speakers in the morning, and in the afternoon we have brainstorming discussions supported by NICE tournament and catamaran tours in the Danube to make sure that it's a very informal scientific discussion. And we right now have a registration open for that. So I just wanted to mention this. Finally, all the scripts and tools that I mentioned are available in our GitHub page or in our website. So feel free to use them. And if you have any questions, and then you can email me on this address. And looking forward to any questions for that, or any problems if you may experience. Hopefully not, but if you have them, looking forward to feedback. Thank you.
Thank you very much, Tamas, for this very interesting presentation, and also for staying in time. I think it was very nice. Hopefully we will have a few questions later. Now we move to the final speaker. This case is Professor George Spiro from Cyprus, and he's going to give an example about how to use this systems medicine approach to a specific disease, Alzheimer's disease, and drug repurposing. Please, George, whenever you want.
Thank you, Antonio. Good afternoon. It is a great challenge to talk after Harold and Tamas. I will try to give you some interesting information also. Let me share my screen. I suppose that you can see now the full screen.
Yes.
Yes,
okay.
we can.
Yes, thank you. So actually, I'm a newcomer in NRF2 biology and Redox biology as well, thanks to Antonio and thanks to this cost action that I am participating. So I proposed that my lecture would split to 2 parts. In the first part, I will talk to you about some concepts that we are developing in our group that are network-based bioinformatics approaches. And the second part is the example that Antonio mentioned related to NRF2 and Alzheimer's drug repurposing. A few words related to our group. This group was, let's say, initiated through the Bioinformatics European Research Area Chair funding in Cyprus, and then it's transformed to a permanent department in the Cyprus Institute of Neurology and Genetics. And to make the long story short, we like to work with networks and to see the systemic approach of the bioinformatics analytics. So we try to perform networks at each layer of omics and then integrate the layers to generate descriptive profiles, understanding mechanisms, and perhaps highlight candidate biomarkers and repurposed drugs. So in the first part, I will talk to you about some concepts that we are developing from these network-based bioinformatics approaches. The first one, I'm starting with pathway analysis. After the very nice introduction and highlighting points made by Harald, I can say that when we are dealing with standard pathway analysis, we have some pathways at hand finally, and many of them, they are not consistent with the initial hypothesis that we had when we were investigating the phenomenon. So what we tried to propose is to map the initial pathway findings to a pathway-to-pathway reference network that can be generated through various databases like KEGG, Reactome, Wiki Pathways, etc. So we are generating a pathway-to-pathway reference network, and then what we are proposing is to map our findings each time to this network. When mapping, we will realize that some of our pathways, they are not connected like these ones. So we are asking a simple question. Are there any other pathways, or what is the minimum required number of pathways to achieve connectivity between all the pathways in the list? And this is done and implemented through a web-based bioinformatic tool that we have published, and it is called Pathway Connector, that takes as input either genes or pathways, and then maps the pathways on the selected reference pathway-to-pathway network, finds the required pathways that were missing in order to achieve connectivity between the pathways that we have, and then we can also proceed with clustering on the pathway-to-pathway space in order to find the so-called connected functional stories, as you can see here, or as you can see here in a work that we did based on the questions related to spastic ataxia and to Huntington's disease. Another concept that we try to develop more is network-based data integration, where we would like to integrate data coming from omics or by other knowledge from existing databases, and this data could contribute to either the nodal content and the edge content between two genes each time, generating a synthetic gene-to-gene network. So we propose that there is a way, a mathematical way, to generate a synthetic gene-to-gene network where there is nodal content and sub-edge resolution that comes from the contribution of the omics or the other existing information. And what can we do with this gene-to-gene network that carries all this information? Actually, we can use it as a map in random walks, or better, on informed walks on various other networks. So, for example, if we have millions of walkers going randomly or informed, let's say, on a pathway-to-pathway network, by having at hand a map, that this map is the integrated network from all the existing information that we have and the omics that we have, then our agents, they are guided to walk in an informed way in our network. So after millions of visits, if we ask what are the overrepresented and overvisited regions of this network, we will conclude with several modules or several communities, and we expect that these overvisited communities, they are connected and they are related to the map that was guiding our agents. And this map is related to the information and the omics that are also related to the disease that we are investigating. And this concept, we had the opportunity to apply it to the, let's say, emergent days of COVID, where we tried to combine the information that we had at hand at that time, and we generated a synthetic gene-to-gene network. This is the one. And from there, we managed to perform random walks, actually informed walks with our generated network map in order to highlight communities of pathways that could be, and we believe that we have evidence that they are related to COVID-19. And further, this information was used to highlight our work on drug repurposing against COVID-19. Another concept that in the direction that we try to find good markers that should be more exploited, either on the discovery of the underlying molecular mechanisms or on the drug repurposing direction, is the work with the monotonicity in gene expression. So in this work, we collected gene expression profiles from colon cancer stages, and we found the genes that were consistently overexpressed across the 4 stages of colon cancer or underexpressed consistently under the 4 stages of colon cancer. So we highlighted and we established, let's say, the term of monotonically expressed genes, and furthermore, the term of monotonically enriched pathways that are related to these genes, because we believe that this consistency is highly correlated with the disease progression. And from there, we managed to find what are the drugs that are capable to reverse this monotonicity, and what are the pathways that they are targeting. So we proceeded with a study trying to find good candidate repurposed drugs that could reverse the colon cancer progression. As Harald said, talking about the phenotypes and the very important contribution that they have behind the names of the diseases, a way that we went in this publication trying to concentrate in a basic phenotype in this publication was the fibrosis, was the following. We tried to collect all the available data sets from Gene Expression Omnipass regarding transcriptomics from 13 diseases that they are fibrotic, of fibrotic type. So we analyzed them separately, and then we tried to generate networks, disease-to-disease networks. Actually, this is the fibrotic module where we tried to connect these fibrotic diseases between them based on several questions. As you know, a network is based on a specific question of relationship. So if we change the question, we keep the nodes, the network changes. So several questions that we said were, can we connect the diseases based on their common overexpressed or common underexpressed genes? Can we connect them based on the common repurposed drugs that we found, etc.? And finally, in this direction of concepts, working on concepts with bioinformatics approaches, I would like to say a few words on this. We are working on the so-called network rewiring. We try to establish networks at each stage of a disease, and then we try to observe the changes between the networks across the stages. So we try to find whether some of the nodes, they are changing friends, or they are changing the intensity of their friendship. Actually, we are investigating another type of biomarker, that it is not a molecular biomarker, but it is a relationship. So we are looking for relationships as biomarkers as well. So by closing this, this first description, I would like to say a few words on the drug repurposing part. The mainstream in the drug repurposing part is to work with transcriptomics data and to find drugs that they can reverse the transcriptomic profile of the disease or of the condition that we are working. But again, we are ending up with lists that we need to make them shorter and to be able to believe to their ranking. So we are working in our group to develop methods that they are using functional information and structural information to re-rank the initial ranked list of candidate drugs in order to be closer to the disease that we are looking. So initially we developed a tool that is called Codres. It's a web tool that can perform this re-ranking based on functional and structural criteria. And then we proceeded more in a publication that tries to propose a new re-ranking scoring with a case study on Alzheimer's disease and especially on the stages of the Alzheimer's disease. So what we are proposing here is to not only exploit the functional knowledge, that is the pathways that are targeted from the drugs that are repurposed, but also to devise a score that is based on how similar are the candidate drugs that we have found with the already known failed drugs or the drugs that are currently on clinical trials or with drugs that are already approved. So we devised a score with penalties and let's say credits on this information. And we proposed the scheme of the drug disease cookie, where the drug disease cookie contains the known information about the disease and the drugs that are related to this disease. So the failed drugs, the FDA approved drugs, the drugs in clinical trials, the drug targets and the pathways. And all around this, we are building a scoring scheme that exploits the initial scoring from the Codres tool that I presented before, and also other scores like the score from the structural similarity with the failed and the clinical trials, and also other scores. In our case, also, we have put the score that relates a prediction with the blood-brain barrier permeability. And based on this scoring, we were able to propose and shortlist several drugs per stage and to find the pathways. Here is the pathway network, where we can see the various colors refer to the various stages of the Alzheimer's, and we can see which pathways they are targeted or they are related to drugs that they are targeting to each stage of Alzheimer's. Closing this part, I will say a few words with regarding a recent work that we called it Dreamocracy. It is on a preprint level up to now, where we are asking, okay, there are several works, actually a lot, that they are proposing drug lists against the disease, or we have lists that they contain approved drugs or repurposed drugs or drugs on clinical trials, etc., etc. Can we capitalize on these prior drug discovery efforts in order to generate a reference? So we devised a method on how to integrate all this knowledge that comes from selected directions, for example, computational repurposed drugs or clinical trials based drugs or experimental drugs, etc., to integrate a matrix where we say, okay, by adding all this information, can we find the frequencies and the overrepresentation of the modes of action and the pathways targeted and the initial indications of all these drugs And if yes, then we can have a matrix as a reference point, and then we can ask this system about a new drug, a drug that we have interest, or several drugs of interest. And then we can say, okay, I have a drug with a specific profile, with a specific known mode or modes of action, an initial indication, and known pathways that it is targeting them. So if I will generate this vector for my drug, then can I find correlation with the reference matrix that I have generated and see what are the most favorable diseases that could be, this drug could be repurposed against them. So this is a concept that we are developing more and more, and we hope that it will help in the drug discovery efforts. Okay, I am switching to the small example. As I said, we are new in the NRF2 research, so I will tell you a short story that it is the work of Dr. Marilena Bourdakou. Marilena is a member of our group, and we work together with Antonio, trying to see if we can say something on the drug repurposing direction, focusing on NRF2 neighborhood. So what we did is that we took the NRF2 neighborhood as presented in a paper of from Antonio and Harold, and this paper and this network builds upon also the NRF2 network that Thomas presented, and we collected these players from the NRF2 neighborhood, and what we did is that we asked the gene expression atlas at EBI specifically for Alzheimer's to say whether we have known expressions, actually differential expression for these players in this repository. So from the almost 230 genes, 80 were found as differentially expressed in 6 brain areas from Alzheimer's disease patients compared to normal individuals. And from these genes, 47 were found as underexpressed and 33 as overexpressed. Here we can see the overexpressed, differential expressed genes in red and the underexpressed with green color. Then we went to a very nice tool from the Mayan lab, the drug gene badger, that gives you the opportunity to ask per single gene to find drugs that they are found experimentally to reverse or to promote this expression profile. So we used several parameters to be sure that we are statistically correct and with correct fold chains. And then we highlighted 43 out 47 underexpressed genes and 29 out of 33 overexpressed genes with proper drugs that could reverse the expression profile related to Alzheimer's. And after putting a filter, by selecting the candidate drugs that they are reversing the expression of at least half of our genes, we ended up with finally 34 unique candidate repurposed drugs. And here are the candidate repurposed drugs and their targets and how these targets, they are found to be overexpressed, underexpressed in the expression atlas of EBI related to Alzheimer's. We used this information, we used the targets of these drugs, and also we used other information coming from papers that are proposing NRF2 related drugs from clinical trials or related to Alzheimer's from in vivo models. And we generated pathways and we tried to find commonalities between the pathways targeted by our proposed drugs and the other pathways that were targeted by drugs that are found by others. So here we can see common pathways with NRF2 related drugs in AD clinical trials are represented in red. Common pathways with NRF2 related drugs in other clinical trials, but with positive effects in AD in vivo models with a star, and pathways that were also found as significantly enriched from the pathway analysis of the NRF2 related differentially expressed genes with a rectangular border. We did a similar analysis, but in a higher resolution with modes of actions. And again, we found interesting commonalities. I will say a few words in the computationally validity checks, and I'm talking only about computational validity and not experimental. So when we try to find whether our findings are structurally diverse, we found a very good structural diversity between the findings that we have and the other drugs that were listed in the bibliography. And also, we found a good commonality between the pathways that we found and the pathways that are related to drugs found by others, both in a pathway level and in the mode of action level. And last but not least, this part here, where we try to find the association of our proposed drugs with several diseases, and then we asked whether these diseases, they are the NRF2 related diseases coming from the paper of Antonio and Harald. Finally, we concluded with several very interesting observations like this. This analysis resulted in 13 repurposed drugs out of the 34 that are associated with the NRF2 related diseases. And here we can see in bold several drugs where I will now highlight them. So we are concluding with curcumin, trichostatin A, panobinostat, and the other two, where we can say that 3 of them, they are A-stack inhibitors, and we believe that as we can find from the literature, they are very interesting to be more investigated against Alzheimer's. So, concluding, I can say that our methods, they can only show a direction or propose candidate repurposed drugs. So, it is part of the experimentalists to believe some of our findings and then to try to focus more and to put experimental effort to highlight and verify whether these findings are true or not. I would like to thank my group and previous members as well, and I would like to thank you all for your attention.
Thank you very much, George, for this very inspiring talk. I think it was really very interesting to see how things are moving along, you know, just coming from an idea, from a database, how this can be built into drugs that are already in the shelves and can eventually be used, can be given a new life in different diseases. So thank you. So we are going to start now with the questions. I don't see any specific question in the chat. So please, if you are welcome to write your questions in the chat in case that you have any. And well, eventually, if you are not feeling very comfortable with writing, eventually you can also raise your hand and I will see if I can see it, I can give you the word. But maybe while you are thinking about this, maybe I can start with a couple of questions or maybe just some summary of the 3 very nice talks that we had today. First, from Harald, we learned at least 3 points. One is that reactive oxygen species are not always the bad guys. And I think this is interesting because, like Harald said, we know that there are enzymes whose only role is to generate reactive oxygen species. So maybe, Harold, you might want to comment on the overstated use or the overuse of the word oxidative stress.
Yeah,
This is easy for you, I know.
it's a bit of a sensitive issue with some of those groups that have been using and establishing it for many, many years. So I think we have to, it's certainly a very important signaling concept, but I think we have to approach it like we approach all other pharmacological targets, namely by the proteins that either generate them, that are the targets of them, or that metabolize them. And the message was that in different diseases, different proteins, different generators of ROS will be important and so forth. And we have to identify those proteins and leave the other sources and targets of ROS that are not involved in that disease phenotype, leave them, if possible, untouched and focus only on the disease-relevant ones.
Do you think this is the reason why antioxidant therapy has never been able to, to provide any really good data?
Yeah. In general, I find it very difficult to imagine that you should take some compounds and basically the entire redox potential of your body should be changed. I think that's a bit unlikely. We are not a battery. So, but also the potential is, of course, that let's assume that some of these compounds really are effective and in your whole body scavenge ROS. They may actually then interfere negatively with some essential functions of ROS. So you may have had some benefits, some unwanted effects, and the net effect was neutral. That may also be an explanation.
Okay, and also in a related question, I was really very surprised by the wonderful data that you have generated with NOX5. I think it is amazing that NOX5 could be a hub in several diseases, and hopefully you will be lucky with your cardiovascular trial.
Yeah.
And my question here is related to the crystal. NOXes have already been crystallized, and I wonder if you have looked at the docking and molecular dynamics of specific drugs that could be just interacting with NOX5 and not with other NOXes.
No, not yet. We're not very successful there. This perfenazine seems to be a rather general NOX inhibitor, but we use it in ischemic stroke. So there you basically treat acutely for 12 hours maximally. And there it probably doesn't matter so much whether you hit also some other NOXes that you don't want to touch. It's basically saving brain, making the patient survive, and there are some possible side effects of other NOXes are not so relevant. I would not consider perfenazine, for instance, as a drug for any chronic indication. There we would need to find other inhibitors. So far, we haven't found them. There is, I thought, maybe one figure that I didn't show, and because George was also talking so intensively about drug repurposing, why it has so much potential is because a beta blocker does not only bind to beta receptors, it binds in average to 30 other proteins. And there's a very nice study that for me was an eye-opener with respect to drug repurposing. So in this study, over 400 FDA-approved drugs were modeled on the then known protein structures. And you can see there are some drugs that really bind only to one or two molecules, but there are also a lot of drugs that bind to basically hundreds of proteins. In average, a small molecule on the market, FDA approved, binds to 32 other proteins. So we have to think not when a drug binds to a protein of that protein, but of the pocket that's shown here, of the pocket to which that small molecule binds. So, and there's another sort of field in cheminformatics which try to estimate how many of these different pockets actually exist in all our proteins. Of course, in a moment, I would say it's an estimate, but one estimate is that there are only 1,400 different pockets on all our proteins to which a small molecule can bind. An antibody can also probably bind to an outside protein sequence, but small molecules need a kind of deeper or flatter pocket to bind, which means if you have only 1,400 pockets, you can maximally have 1,400 different drugs. The next drug will already bind to a pocket for which we have already a drug. So there is a huge potential for drug repurposing, and the number of all drugs that we may ever need is actually finite. So we will not need to eternally find new drugs.
Yes, but also I think you are talking about mechanisms, but remember, many of these drugs have specific pharmacokinetic profiles.
Yeah, of course. That's sometimes a drug may have an unfavorable pharmacokinetic, and then you may need to find a drug with a longer half-life or shorter half-life. Yeah, sure, absolutely.
All right, thank you. So I still don't see anybody willing to make questions. I guess we are shy. Sorry for that.
Isn't there a drug here, a question from Tamas?
From Tamas? Oh, yes.
No, I got the comment. Yeah, I got the comment.
Oh, you answered the comment already, or?
No, I haven't. It was posted as in targeted to me, which was a really interesting comment. Was on, and thanks for that. Is NRF2 in IBD, was the immunological aspects of involving chemokines and regulation of NRF2, or is there any crosstalk of the immunological function of NRF2? Or is it mostly related to the cell protection role of NRF2 that is apparent? And on one side it's a comment, but I think it's a very correct one. And this is why this whole field is quite exciting, I think, to identify these crosstalks and comments. And as I was just reading this, I just quickly opened the figure we did before. So this was basically a functional overlap analysis about those protein interactors that are working with NRF2. And I just wanted to point out this in the middle. So these are the immune system related protein interactors, and then there are some, then they overlap with the others. So on one side, from the 27 immune system related ones, indeed there are lots of signaling related ones, and indeed lots of them are related to cytokine or chemokine signaling. For me, what was a really interesting finding here, and then I'm quite interested, is the wound healing part. So how proteins that are relevant in wound healing are there, and then their overlap, for example, with immune-related pathways of chemokines is smaller, like 2/3 of all the proteins. And this could be quite interesting when we are targeting the immune system and how it may affect NRF2's role in wound healing. So just a very quick response to this very, very good comment.
Thank you. So
Thank you.
In my opinion, the most robust role of NRF2, of course, is in redox regulation, but also in inflammation. I think this is really very, very clear. And this leads me to the question that I wanted to make to Tamas, which is related to the quality of the databases. Because, you know, we say that NRF2 is very important in inflammation because there are a lot of papers saying that, but maybe it is very important in something that is, there is marginal now in the literature, and it will become actually the most relevant function. So my question is about the databases. You talked about curating the database, but every time that you curate the database, you are approaching your experimental situation to your idea. So basically, you are reducing the chances of getting new information. So what do you think about the quality of the databases these days?
Thanks. That's a very, very important question. So in general, I think what we always need to acknowledge and keep in my mind is the bias that exists in the literature and in the data sets that are often available. It's often a study-related bias. There is also, for example, any kind of cancer-related proteins have always more interactions than others, and it doesn't mean that that the other proteins don't have that many interactions. It's just that those proteins were more studied. So I think it's important to keep that in mind. There are multiple ways how to work around that. One is actually using multiple resources. So trying to look at different resources that were compiled with different type of protocols. And I didn't mention in my talk, but this Omnipass resource we developed, the Nature Methods paper that we published in 2016 about Omnipass, it's actually not the paper itself that is very good there, and the important is the supplementary material. It's a long benchmarking analysis of more than 30 different resources, and we compare them based on various parameters, how good they are and when they are good. And basically the main message of that is that it's not about that there is this one single resource that is great. It's about the use case you have and what you would like to do. And in case of, for example, what we did with functional analysis, in case of antioxidative response, NRF2 had higher percentage of relevant proteins than other resources, but in case of obviously other resources. And if you are looking for a more comprehensive view, it's about combining these network resources. So I think that's why data integration is quite relevant, and you don't have to do that. Others have already done it. You just need to make sure that you are working with the relevant resources.
Very interesting. And also, I think I want to thank you for joining our cost action, because we have several deliverables, and the NRF2 OMA and also the microRNA data is going to be a wonderful deliverable.
No, thank you for that, and I'm really looking forward to working with many of you on the call.
And okay, while people decide to make some questions, I want to go now to George, if you allow me. So George, I know part of the results that you have presented today because I am co-author of the nice study that you already discussed about Alzheimer's. But something that is interesting in this study is that you brought your most interesting drug, which is present in something like 19 of your it was very evident in your analysis. It scored 19, which is the highest one. Turns out to be curcumin, and curcumin is it's very popular nutraceutical. You can find it in many pharmaceutical places. There are a lot of clinical trials going on, but to my knowledge, it has never been used conclusively in a clinical trial. So I don't know whether this kind of approach, if this is happening with curcumin, imagine what is going to happen when we go to the other compounds we don't score so high. So my question is, how do you see this? If you had €1 million or dollars, or I don't know the Cyprus coin at this time, would you put it in one of these compounds?
Antonio, I don't know if I am the best one to give this answer, but from the bioinformatics perspective, I tend to believe to these findings, these highlighted findings. So if I had the money and I was the one that I would take the decision, yes, the answer would be yes, because sometimes the bioinformatics give the opportunity of agnostic approaches to highlight some compounds or entities that are not directly related to the condition that we are looking, or directly related to the a priori knowledge that we have. This is let's say, the beauty of the network analytics that can provide inference and give you some, let's say, agnostic ways to highlight unexpected findings. So this is my explanation, and this is also my answer. Of course, it is an easy answer because I have not the money. But yes.
If you allow me to answer myself, what we can do, and I just suggest that we can do it together, is to take those 3, 4, 5 compounds and put them in the preclinical models of Alzheimer's disease and see what happens. If they work in the preclinical model, well, maybe there is a chance to go to the clinical side very quickly because they are already in the market.
Yes, and we have, let's say, the opportunity to start getting in some experimental verifications of our findings. For example, in the work against fibrosis, an experimental group tried the highlighted by us niclosamide, and there are very interesting preliminary results, not only from this group, but from other groups as well. And also we have some very, very preliminary good results in vitro assays with the last work that we performed with Alzheimer's disease in staging. So yes, I agree with you. It would be very nice to set up a small experimental procedure to work on this.
Okay, Harald, you have a question in the chat. Would you want to make it open?
Sure. So yeah, I read the question. Yeah, Alzheimer, we have actually no indication so far. You know, I don't like the term oxidative stress. Yeah, so we don't have indications that there's a signaling pathway that generates amounts of ROS that become directly neurotoxic. But what we see is that this NOX5 interference with neuroprotective cyclic GMP could actually be relevant for what I would consider a gray zone between vascular dementia and Alzheimer. Also, Alzheimer is probably a mixture of different pathomechanisms, and so that may well be. And the wild type protein, the amyloid wild type protein, precursor protein, that is actually anti-apoptotic. So I don't think anti-apoptosis is a mechanism that induces Alzheimer. In the opposite, it protects from Alzheimer. And there are a couple of groups who believe that it's not always the accumulation of amyloid we need to look at, but also at the reduction of the precursor protein and its physiological anti-apoptotic effects. So it may actually be a proteinopathy rather a proteinopathy.
Yeah, I agree that the amyloid precursor protein is probably the target rather than the A-beta peptide. Actually, yeah. You can clean the brain from the amyloid plaques and still you have dementia. So that's it.
Exactly. Yeah.
All right. So I don't think there are more questions here. Maybe Luciano, would you like to participate in this discussion and maybe close? Luciano? Okay, while we wait for Luciano, any other question? Brigitta?
No, I am reading in the chat. There is another question from Opugunduri.
Yes.
About the curcumin, bioavailability of curcumin, or the more that make it difficult to establish it unless if we can have means of delivering to the target tissue directly. On the other hand, it also has other effects such as inhibition of the glutathione trust. Is a comment about the bioavailability of curcumin. Yes, but I don't know. This is not here the field to.
I think there are several studies trying to change curcumin to something
the structure, but
some people call super curcumin.
Super
in the sense that it is encapsulated and it is somehow modified to be more stable and provide a much better pharmacokinetic profile. But as far as I know, maybe Rao has a different view, and you are welcome to participate, just to open your microphone. But I think, as far as I know, either curcumin or curcumin derivatives have not reached phase 3 clinical trials so far, but I may be wrong.
Yes.
Yeah, thank you for the remarks. I mean, I just wanted to convey the opinion, like why it could have failed is because of the reason that, you know, maybe we are not yet there to deliver it directly to the target tissues. And even though we claim that we made several ways of delivering, we do not have a clear evidence, you know, with the properly levels reported and measured from these target tissues and the amounts that supplies. So I think this is where we are lagging behind and not equipped with to reach that stage of, you know, seeing the efficacy of that molecule. I mean, it's a personal opinion, maybe not scientifically based, but thank you.
The problem is, I mean, we can have many different opinions as long as we don't have robust scientific evidence. So
yeah,
this is the point. All right, so I don't think we have more questions. We can now think about closing this session. I think it is important to thank our three speakers because you really gave us a lot of new information. For many of us, at least for me, systems medicine is very complex. I like to go from the simple things to the complex things, and you go from the complex things to the simple things. So for me, it's quite complicated. Luciano is back. So Luciano, maybe you want to finish, maybe you want also to close this webinar.
No, thank you. Just want to echo what you are saying. Many thanks to the speakers for the excellent talks. Many thanks to you, Antonio, for chairing so well this webinar. Many thanks again to Brigitta, who did a lot of work for the preparation of this, this webinar, and also to the participants. I see still 40 people online, so it was a long session, and I'm happy that most of them remain for the whole event. So many thanks, and see you soon on this website. We are planning other webinars of this high level, and we hope to see all of you again. Thank you.
Bye.
Thank you. Bye-bye.
Thank you. Bye-bye.
Bye-bye.
Thank you.